presenter is Garret Stuber, and like Mark Baer, he's one of the DECODE awardees. He earned his PhD in neurobiology from UNC Chapel Hill and did postdoctoral work at UCSF. He won a number of awards for his research to delineate the neural circuitry that underlies motivated behavioral processes. And he's presently a faculty member in the Department of Psychiatry and the Neuroscience Center at UNC Chapel Hill. We're very pleased to have Professor Stuber with us.
All right, thanks a lot, Mark, and I'd like to thank Inscopics for organizing such an exciting summit today. Today what I thought I would do is share with you some new work from my lab where we're trying to leverage multiple cutting-edge technologies in order to really better understand the importance and necessity of genetically defined cell types within a particular brain region, in this case the extended amygdala, which I'll talk more about in a minute. This project is still what I would consider to be very much a work in progress, but I think that these data that I'll be sharing with you today will really highlight some of the challenges and opportunities that has already been brought up this morning in the panel discussion. So my lab studies the biological basis of motivation. Motivated behaviors such as food seeking, drug taking, escape from threatening stimuli are without a doubt essential for survival. What is also very interesting to me is that how these processes are perturbed or become perturbed within disease states. So in philosophy such as Buddhism, 3 poisons have been attributed to essentially being the root of all suffering. They're listed up here. Ignorance, attachment, and aversion. To me, it's really interesting that 2 of these so-called poisons can be attributed to disruptions in motivation, in particular attachment and aversion. In addition, we know that disruption in neural circuits that control motivation are likely underlying almost all types of neuropsychiatric illnesses. And the ones in particular that my lab focuses on is studying circuitry that controls reward-seeking and addiction behavior, those circuits that we think are important for both adaptively and maladaptively generating anxiety-like behavior, how circuits control or can reduce motivation for natural rewards, which is often seen in many different neuropsychiatric conditions. And as I mentioned, I think that disruptions in circuit function likely underlie many, if not all, forms of mental illness as well as other disease states that we typically aren't necessarily associating with the nervous system, such as obesity. So at the biological level, we know that ancient brain structures are absolutely critical for controlling various types of motivated behavior. Much of my lab's work has focused on the connectivity and function within a number of subcortical structures, including the hypothalamus, the basal forebrain, and ventral midbrain. These structures, like pretty much everywhere else in the nervous system, are incredibly complex and composed of genetically, molecularly defined cell types, many connections from other areas of the brain, et cetera, that are all seemingly interarranged in a heterogeneous fashion. So we think that these cell groups within these subcortical structures are some of the most critical regulators of different types of motivated behaviors. However, we know that these don't function in isolation, and of course they're a part of a much larger circuit. So one example of this is that we know that sensory and higher cortical areas are directly projecting to and likely interface or act to orchestrate the activity of these subcortical targets. Additionally, something we've already heard a little about this morning is that intrinsic signals from the body, such as circulating hormones, are also powerful biological signals that can act to shape the activity or function of these circuits at various points in time or under particular conditions. And it's really these 3 aspects of circuit function with respect to motivated behavior that my lab's interested in. And so we think that these signals that are sort of delivered to these important subcortical structures, which I'll talk more about in a minute, are then ultimately conveyed to other brain areas, basal ganglia, brainstem, et cetera, that are going to be driving motoric output that we can actually observe in animals. So what I would like to tell you about today is some work that actually started in my lab about 4 or 5 years ago now. And it really is a continuation of this project, which we published a few years ago back in 2013, where we were able to identify different cell types within the bed nucleus of the stria terminalis, or BNST, that can drive a variety of motivated behavioral states. This initial work was done by Josh Jennings and Dennis Sparta when they were a graduate student and postdoc in my lab, and they've since moved on. What was really fascinating about this study is I think it was one of the first to really start examining the cell types within this particular brain region and the functional consequences of their manipulation. And just to give you a quick summary, what we saw was that we were able to target a subpopulation of neurons, these glutamatergic neurons, using sort of contemporary approaches in mouse genetics. When we optogenetically activated these cells, we saw that we could increase anxiety-like behavior in a number of different mouse behavioral assays, and we could also promote aversion or avoidance of stimulation of these cells. Now, what was really interesting is that we also did pretty much the same experiments, but in a different mouse line, where instead of targeting glutamatergic neurons, we were targeting GABAergic neurons in the BNST that were projecting, in this case, to the VTA in both cases. But when we did these manipulations on these cells in the same area, we saw that we could produce almost opposite behavioral results. We would see that optogenetic stimulation of these cells reduced anxiety-like behavior and promoted reward. This was some of the first evidence, at least to us, that these different cell types could have very dramatic differences in terms of their functional behavioral readouts when we manipulate these things. One thing that sort of we started to think about more when we were finishing this study is that, okay, well, these are ways we can manipulate different cell populations, but these are still very broad spectrum cell targeting strategies. We're still essentially targeting all glutamatergic neurons or all GABA neurons within the structure. And so after our 2013 paper, we began to search for other genes which may be enriched in these structures, such as the BNST, that could serve potentially as genetic handles or entry points, which will allow us to tap in and modulate and study more precise cell types. So initially, this was done by looking at gene expression data from resources such as the Allen Brain Atlas, and then we would follow up on a variety of markers using in situ hybridization and a number of other approaches. And so one marker in particular that we became interested in, again, just from sort of mining these publicly available data sets, was this marker for a gene Prepronociceptin, which is a gene product that is proteolytically cleaved into multiple neuropeptides. I'm not going to be talking about the function of these different neuropeptides today. We know they're important in a variety of behaviors, but again, we're using this simply as a way to, we know that this subpopulation of neurons within the BNST are expressing this gene, PNOC, and we can use this as a way to eventually tap in and modulate and manipulate the activity of these cells. And so we know the neuropeptide serves an important function, such as increasing pain sensitivity, et cetera. Given that we see this sort of sparse but very dense expression of this gene within the BNST, which is sort of here and here, we thought it would be an interesting gene to follow up on and study in more detail. So we did this first by performing some double and triple in situ hybridization experiments. And essentially what we were able to show is that these PNOC expressing neurons within the dorsal BNST represent a very small fraction of the total cells that are there. And one thing I'll mention is that there's these cells the expression of this gene is not only in the BNST, it's throughout the brains, but in this case, we're just studying it within the context of the BNST. And we saw that this was a small population of neurons that were largely, almost completely GABAergic. So if we stained for, if we looked at the expression of the PNOC gene and VGAT and VGLUT for markers of glutamatergic and GABAergic neurons, we see that by and large, these are all a select subpopulation of GABAergic neurons. So after we began looking at this in more detail and wanting to study these cells, we teamed up with a few of our collaborators, Michael Brukas at Washington University and Thomas Cash at UNC, and we generated a transgenic mouse that would permit us to target these cells for manipulation. So we made a PNOC iris-Cree knock-in animal. So just as an aside, this is obviously a sort of incredibly laborious and low throughput way for identifying novel neuronal cell types that may be of interest or have therapeutic relevance. And there's clear limitations with it, and these are things that I hope we can talk more about later today, such as it doesn't tell us if these cells that we're targeting or manipulating are also expressing other important genetic markers that maybe other people are studying in different contexts, for example. So I hope we can come back to this at the end of my talk. But so we made this animal, and then using standard optogenetic approaches and viral targeting approaches, we were able to selectively target these PNOC expressing neurons within the dorsal BNST. So here's an injection of channel rhodopsin conjugated to YFP in a Cree-inducible fashion. You can see sort of very sparse targeting. Under higher magnification images, you can see these cells have very dense dendritic processes all throughout the BNST. And in fact, we noticed that with slice electrophysiological recordings that these cells have a lot of intrinsic local connectivity within the BNST. So if we photostimulate these neurons and make whole cell recordings from either non-PNOC expressing neurons or PNOC expressing neurons, we see that a lot of these cells receive local inhibitory input from these cells. So when we stimulate the fibers, we can see very robust inhibitory currents on neighboring cells. We know that these cells also have distal connections as well. So if we just quantify the amount of fluorescence in the fibers that innervate a number of different brain regions, what we saw was that these PNOC expressing neurons are targeting two areas to a much greater extent than the rest of the areas that they innervate, in particular the medial preoptic area and the medial amygdala. So aside from just sort of characterizing some of the basics of these cells, we were really interested in whether or not these have sort of unique functional encoding properties. And so obviously we need a way to record and monitor the activity of these cells. And so this is where the Inscopic's Invista system came in. So we were able to genetically target the calcium indicator, GCAMP6S, selectively to these PNOC expressing neurons. And then using the miniature microscope, essentially image the activity of these neurons that are located about 5 millimeters deep into brain tissue. And so this was work that was done, this is sort of, looks a little bit weird on the screen, but what we saw was that we can clearly monitor the activity of these cells. And this was work that was done by two very talented postdocs in my lab, Hiroshi Nomura and Jose Rodriguez Womagera, and they've been jointly working on this. In addition to that, we've been working with other groups to really develop novel data analysis methods on this epifluorescent calcium imaging data. And I'd just like to put a shout out to our collaborators, Liam Paninsky and Pengcheng Zhao, who have been working with us on automated ways of extracting the calcium signals from these data sets in a lot of different brain regions. So one thing that we've sort of found somewhat challenging is that the actual analysis of the data is very laborious and difficult to do, and it requires the help of not only neuroscientists who are working on these experiments, but also people in computer science and people with other data analytics expertise who could really leverage their knowledge to help better understand these data. So we studied the activity of these cells first within the context of standard mouse anxiety-like behavior assays. So this is an elevated plus maze. It's a standard behavioral test for anxiety. Typically what you do is quantify the amount of time the animal spends in these open arms relative to the closed arms. And it's thought that these open elevated areas are more anxiogenic to the animals compared to the closed arms. And so what we did was to combine this with calcium imaging to monitor the activity of these PNOC expressing neurons. And so what we saw was, as expected, you see that the animals spend substantially less amount of time in the closed arms of the elevated plus maze relative, or yeah, substantially less time in the open arms relative to the closed arms, which is what we would expect. And if we look at the activity of these PNOC expressing neurons in the dorsal BNST, what we noticed was that if we restrict our analysis to times when the animal is transitioning from the closed arm to an open arm, seen here, we could see a very substantial increase in the activity of many of these PNOC expressing neurons in the BNST. Conversely, when we looked at the other transition, so the open to closed arm transition, we also saw a reduction in activity, and this is just summarized here. So it seems when the animal transitions to this anxiogenic environment that this is producing changes in activity of these neurons. We've also developed some other analytical approaches, such as a GLM analysis, which allows us to correlate the activity of many individual neurons with a variety of different behavioral states. And so what you can see here is just an example of some of those states that we can look at, such as distance from an anxiogenic environment. And we can just keep adding new behavioral variables if we, depending on what we're actually interested in. But one of the most striking things here was that there's a strong positive correlation in many of the cells with respect to how far the animal is from the open arms or the anxiety-provoking environment in the elevated plus maze. We also wanted to look at the activity of these cells in other anxiety-provoking situations. So we've wanted to use and expand from just using the elevated plus maze to look at this to other tasks. And so what we chose to look at was exposing the animals to a threatening odor, so this or an appetitive odor. So TMT is a component of fox urine, and it's thought to be a anxiogenic or threatening stimulus to mice. And so we could simply place this odor source within their home cage and quantify their behavioral state with respect to where they are in location to that odor source. And sort of as a control, we've also used an appetitive odor, peanut oil, which is something that the animals, if they've had experience with it, will approach and try to consume often. So we can sort of use this and compare the activity dynamics between these cells to these different odor sources. Behaviorally, you can see that they're very different. So with TMT exposure, the animal spends very little time interacting with the odor source within the chamber. However, when you expose the animals to the peanut oil, which again is appetitive, you can see that they show a significant increase in the amount of time that they're interacting with it. So we looked at the response patterns of these PNOC expressing neurons in the dorsal BNST. This was a data set where we recorded about 160-something neurons. And this is their response pattern to water presentation. So this is just our sort of control solution that we would put in the cage. And you can see that there's not really much activity to that. But what you can see is that when we do the same experiment and this time expose the animals to the predator odor, TMT, you can see that there's a robust recruitment and activation in many, but not all of these nociceptin expressing neurons in the BNST. When we do this comparison for a subset of neurons, that were exposed to the peanut oil versus TMT. What's kind of interesting is that we see a response that's larger than what we see with just water exposure. It is evoking some activity, and again, the animals are approaching the stimulus in this case, but still the TMT response is much more, it's a much larger response to this aversive odor. So again, we can run the same GLM analysis and correlate the activity of a number of neurons with a variety of different behavioral states. And one that is probably the most interesting is the number of neurons that are correlated from the distance of the aversive odor source compared to the number of cells that are correlated with the distance from the appetitive odor source. So we can see that there's a strong negative correlation, meaning that the cells are more active if the animals are closer to the odor source with TMT that doesn't exist for the peanut oil exposure. So in addition, we've done optogenetic manipulations of these neurons, so we can put channel rhodopsin or halorhodopsin to optogenetically stimulate or inhibit these nociceptin expressing neurons in the dorsal BNST. And we did this in restricted time epochs where we would stimulate or inhibit the cells for a short period of time during the elevated plus maze test. So you can see this is the location of a representative animal in the closed arms and open arms before the photostimulation. So this is channel adoption mediated stimulation of these cells. But when we can stimulate these cells, that this reduces the amount of time that the animals spend in the open arm of the elevated plus maze. Again, supporting that if we drive the activity of these cells, this produces anxiety-like behavior. So we did the same experiments in a different group of animals where we suppressed the activity of these neurons, and essentially we saw the opposite result. We would increase the amount of time spent in the open arms of the elevated plus maze if we silenced these PNOC expressing neurons in the dorsal BNST. So just to summarize what I shared with you today, I showed that these BNST nociceptin expressing neurons are a small cluster or subset of GABAergic neurons that are located within the dorsal BNST largely and display local and distal synaptic connectivity, and we're interested in following this up still in more detail. These cells show enhanced activity as mice transition to anxiogenic or anxiety-provoking environments. So we saw that they'll show that robust increase when they enter the open arms of the elevated plus maze or in response to TMT, the predator odor. And that bulk optogenetic stimulation can also produce anxiety-like behavior, and we can reduce this by optogenetic inhibition of these neurons. And so I think that's interesting, and the optogenetics and imaging sort of fit together, which is nice. But one thing that I want to end my talk on is really this question, is how can we transition from the circuit level of experimentation where we identify, say, a genetically defined cell type within a particular brain region and characterize its importance or relevance for a number of behaviors. How do we transition from those experiments to identifying potential therapeutic targets within these circuits or cell types? Obviously, we're not probably going to be doing optogenetics or even chemogenetics in humans anytime soon. So it's important that we really think about other ways that we can leverage these data in order to build new therapeutic opportunities. And so the one way that we're pursuing this is using using single-cell sequencing technologies where we can go in, take tissue that we know contains the cells we're interested in, dissociate them, and then capture the single-cell transcriptomes in a very high-throughput fashion. So we can profile the transcriptional landscape of thousands or hundreds of thousands of neurons from many different animals, and then go back in and look at, say, query the data set for just the PNOC expressing neurons and look to see what other genes that they might have expressed that would be sort of unique to those cell types. And so this is obviously gaining a lot of traction and interest in these approaches. This is just one approach that we're using, DropSeq, which was published by Evan McCosco and Steve McCarroll last year, and it's working really well for us to do these studies in many subcortical circuits. And then finally, I would just like to end on that resources are now becoming available to start processing these data. So here's two examples. The Allen Brain Atlas has their single-cell sequencing porthole, or the Broad has their single-cell sequencing portal, and the Allen Institute also has something very similar. So I think these resources are going to become very important for mining these very rich and dense data sets for novel therapeutic targets. And I'd just like to also add that I think we need resources analogous to this for the functional data sets, such as calcium imaging. Right now, individual labs are doing their experiments, seeing things, but the data is clearly not being leveraged to the extent that it could be. And more so, I think that it would be great if we could think a little bit about how we could link up these different resources to each other. So if we have a database for, say, functional imaging of neural circuits, how can we talk to these databases that are going to have lots and lots of single-cell sequencing data? So with that, I'd just like to thank everybody in my lab who contributed to these studies. Again, Hiroshi Nomura and Jose Rodriguez Romaguera were the two co-first authors on this study, and they had a lot of help from a number of other people in the lab, other postdocs, Jenna, my MD PhD student Randall, Elliot also helped out. And I'd also like to acknowledge our collaborators, Liam Pandinsky, Mark Zilka at UNC, who's been working on the DropSeq stuff with us a lot, Michael Brukas and Tom Cash for generating the PNOC pre-mouse with us, as well as our sources of funding. Thank you very much.
Hi, David Anderson from Caltech. Very nice talk, Garret.
Thanks.
I was wondering how you distinguish whether the negative correlation between BNSTPNOC activity and TMT reflects level of anxiety or is rather a sensory representation of the olfactory stimulus, which increases a function of concentration as the animal is closer to the stimulus, especially given that those cells are activated by peanut oil as well.
Yeah, no, it's a great question. And one thing that I wanted to mention in my talk, but I didn't sort of skipped over, is that we've sort of put this in the context of anxiety-like behavior, and yes, that has probably an important component, maybe relevant to things we're talking about in humans, but I just want to put a cautionary note that a lot of these behavioral tasks that we're using to monitor behavior in rodents are open to interpretation, even though these tasks are sort of designed to be the gold standard for testing, say, anxiety-like behavior. There's clearly other things that the animals are attending to and doing that may not be related. So I think your point on whether the olfactory system or olfactory memories are coming into play is a point well taken. It's something that we cannot completely dissociate given this task that we're doing. However, we have other tasks such as that are much more behaviorally rigorous, such as in head-fixed animals, where we can dose different concentrations of odors that will lend itself better to do those types of studies.